The broadcast is now starting. All attendees are in listen-only mode.
Hello, I'm Catherine Poole, President and founder of the Melanoma International Foundation. We are pleased to bring you this cutting-edge webinar today on the TILS therapy, also known as the adoptive cell therapy. Please join us in welcoming Jeff Weber, Director of the Donald Adams Melanoma Research Center at Moffitt Cancer Center. Thank you for joining us, Jeff, today.
Oh, thanks very much for the invitation. Good afternoon, everyone. We're going to talk today about the immune system and how it can be used to treat melanoma. And we're going to talk about something called adoptive cell therapy. When you hear the word TIL, by the way, TIL stands for tumor infiltrating lymphocyte, which, as you'll hear in a couple of minutes, is a relatively novel way of attempting to treat melanoma, which has met with some success recently. If you look at that first slide, you'll see on the left a pretty ugly-looking primary melanoma of the skin. What's interesting about it is that it has areas that are dark and areas that are light, and an area in the upper left corner that's almost moth-eaten. That's because it's had spontaneous regression. Spontaneous regression occurs when the melanoma itself can actually generate an immune response, and there's an attempt by the body's immune system to cause the tumor to disappear. Obviously, in this case, it didn't work completely, but if you look at the right-hand panel, it turns out that the melanomas, which are stained in red and which have these ugly blue nuclei, have lots of little dots, very dark little dots interspersed. And it turns out that a lot of those little dots are actually immune cells. And if you look at that lower panel on the right, the melanoma cells are stained in light blue, but in dark blue, or I should say in dark brown, are stained a lot of the immune cells, suggesting that this melanoma is actually chock full of immune cells. And it's the hope that you can use those immune cells to cause the tumor to regress that is the subject of this talk. So if we go to the next slide. The basic idea is that it is possible that the immune system could seek out and destroy the cancer cells. As I showed you in the previous slide, some melanomas spontaneously disappear, or at least try to shrink. A lot of times patients with melanoma develop depigmentation or vitiligo, and that's an immune reaction against the pigment cells or against the tumor. And there are a lot of strategies that our immune system, which frankly evolved to combat infection, they can be used to fight melanoma and other cancers. And there are a wide variety of immunotherapies that have been tested that either boost or induce natural immunity against melanoma. And two of them actually were approved by the Food and Drug Administration. So if you look at the next slide, it shows you that there's some pretty unique features of the immune system. It's a biologic system. It's a living part of your natural defense. And the immune system has a number of hallmarks. It's very targeted and very specific. It can also be very vigorous. Anyone who's ever had a bad cold knows what a vigorous immune response is. And the immune system has memory. It has the capacity to remember. And if you compare immunotherapies, or at least most of them, to chemotherapies, many of the side effects tend to be minimal compared to chemotherapy itself. You don't see hair loss. You don't see much of an impact on your blood counts. There's no nausea or vomiting. Often you see other side effects that are very different than chemo, but the immune system holds out the hope that if it's very specific, it'll hit the tumor and not the normal tissue. So let's look at the next slide. And those are the players. The tumor cells, the ugly ones, are up at the right. But there are a number of different arms of the immune system. In the lower middle, you have those yellow antibodies, which can specifically seek and bind to the tumor, or can bind to immune cells to promote their activity. You have your killer cells at the lower left, And those little colored dots are essentially the ability to be armed to recognize the tumor. And some of those cells make cytokines, which are chemicals that themselves can either promote the activity of other immune cells or could even kill the tumor cells. So if we go to the next, it shows that immunity generally works well against infection, but the problem is it doesn't often work well against melanoma in the natural state or against cancer in general. And why is that? Well, let's face it, cancer cells are derived from normal cells, and they often differ from the normal only in the expression of normal genes. The other thing is that those cancer cells are nasty. They can evolve quickly to lose the substances that can be recognized by the immune system, which are called antigens. And they're very nasty. They can produce suppressive substances called cytokines and other molecules that can actually decrease immunity. And finally, patients who are sick with cancer who've received prior chemotherapy often don't mount a very good immune response. And in the next, it shows that there are multiple ways that the tumor can actually try to evade the immune system or avoid it. And normally the immune system surveys the body, so you have what we call immune surveillance. But if you hit the next, you'll see there's immune evasion, and you have a very low frequency of tumor cells. And the way one tries to get around that, if you click next, you can have highly functional T cells that tries to get around the first problem. And if you hit next, because you have a low burden of T cells, the only way to get around it is you just get more of them. So let's go to the next slide. And you'll see there are various arms of the immune system. We briefly talked about the antibodies. Those are chemicals. They look like little Y-shaped structures. And they could actually attack the cancer directly, or they can bind to molecules on the immune cells and trigger immune activation. You can actually remove the barriers to the immune response, which is like cutting the brake, or you can bolster immune activation, which is like pressing on the accelerator pedal. Next. And there are a number of these antibodies that exist. We have several immune-stimulating antibodies. I just list them at the top, and those are all in phase 1 trials, the very earliest stage of development. If you go to the bottom where it says blocking negative molecules, those are antibodies that actually cut the brake. And one of them was just approved last year. That's ipilimumab, or the anti-CTLA-4 antibody. There's another promising one called PD-1, and yet another one called PD-L1. And all of those are substances that act as immune brakes. And when you give an antibody, you cut the brake. And those are all in phase 2 trials. Next. Once again, if you look at the players, if you look at those antibodies, you can see that those antibodies can actually affect the killer cells. And those are the cells we're going to talk about in a minute. In addition, the cytokines that we talked about before, if you look at that arrow toward the killer cells, they can also promote the activity of those killer cells. And all these are good things, and we'll talk about how this has been realized. So let's go to the next slide. So what are these T cells? Well, what you do is you could actually remove the patient's own white blood cells, select in the lab the cells that would seem most likely to be the anti-cancer cells. You could arm them for anti-tumor potency and reinfuse them into the patient where these, quote, engineered, unquote, T cells now seek and kill the melanoma. Next. So what is this adoptive cell therapy? Well, if you look at the little diagram, the cartoon at the left, you see the little person. And if you look at the lower right, you actually isolate the tumor. So you isolate the T cells from the tumor, and those are the so-called tumor-infiltrating lymphocytes. Or you actually bleed the patient and isolate some blood cells. And then in vitro, which means outside of the body, or ex vivo, the same term, you enrich them and you expand them. You make them grow. And you use all kinds of different little tricks. You use IL-2 or interleukin-2. The antibody against CD3 called anti-CD3. Feeder cells, which are just normal peripheral blood cells that are radiated. Or you can actually pulse little pieces of tumor-specific protein onto something called a dendritic cell or a DC. And then you grow them to huge numbers outside the body. That's in the green. And then if you look at the little diagram, up on the T-shaped bar is a bag of cells. And you reinfuse the T cells back into the patient's body. And those T cells, like I said, are grown either from the tumor that you digest up or chop up, or from the patient's own peripheral blood. Next. And again, if you look at the left-hand side, what we call active immunotherapy is where you actually inject a vaccine into the patient. And if you look at those little gears, that's designed to show you that the gears of immunity are moving after you inject a vaccine. And the immune cells in the patient that could kill the tumor are the little green guys. And if you immunize somebody long enough and well enough, you'll get lots of little cells that are tumor-specific and kill the tumor. On the right side, totally different story. That's passive immunotherapy. Why is it passive? Because you're actually growing the cells outside the body, and that's those little bottles. All of those are bottles of media with lots of cells that are growing. And you grow them to huge numbers. We're talking 100 billion. That's a big number. And you put them in a Teflon bag, and it's like almost a quart's worth of cells. And you inject those cells back in the body, but you're growing them outside the body. The body itself did not make them grow. That's why it's passive. And that's called adoptive cell therapy, where you grow the cells outside the body and you transfer them back. Next. And again, if you look at the cartoon, you have the cells called the dendritic cells up in the right-hand middle portion. And those dendritic cells get armed with substances that the tumor makes. Those are the little red dots. And eventually they would meet up with the immune cells, those in the lower left-hand corner, and those are the CD4 and the CD8 T cells. And if it's done right, those cells should then leave the area where they're in the lymph node, go to the tumor and kill the tumor. But if you keep hitting the next, you'll see what happens. So let's go next. Unfortunately, those dendritic cells don't work terribly well in the tumor setting. The tumor tends to inactivate them, and they never get to where they need to go. So if you hit next, they disappear. And what we do, if you hit next again, you actually make the cells. If you grow the cells outside the body, instead of depending on the body's own natural defenses, you provide the defenses, and now those transferred cells will migrate to the tumor, hopefully kill the tumor. So let's go next. So what is actually happening when we do this? So how are the TIL cells actually grown? The tumor is actually surgically removed. And it requires a tumor that's probably almost an inch long. You actually physically mince the tumor up into tiny chunks, and you place each little chunk into a well of a plastic sterile dish. You then put the plates or dishes in an incubator at 37 degrees body temperature for up to 3 weeks. The immune cells actually migrate out of the little chunks, settle out on the plastic, and the chunk just sits there. And it eventually becomes a husk because the cells will die because the immune cells, once they escape the suppressive milieu of the tumor chunk, kill the tumor. After about 3 weeks, you then test the cells that escape from the tumor chunk, which have grown in your little liquid media, and you test them for their recognition of tumor. If you start out with 100 chunks, maybe 20 of them might recognize the tumor, and you pool that 20 group, only the ones that could recognize the tumor, and those are the ones you expand to big numbers. So if you go to the next slide, what you do is, after a couple of weeks of expanding the right cells, you harvest them, you collect them. A week, however, before that harvest, the patients are treated with 2 chemotherapy drugs to wipe out the existing white cells temporarily. And then the expanded TIL, or tumor-infiltrating lymphocyte cells, are injected into the veins over about a several-hour period. The next morning, you start interleukin-2 therapy to keep the cells nourished. And then the interleukin-2, the IL-2, is given every 8 hours in the veins until the toxicity is unacceptable. And you stop. The patient recovers, they go home, and 6 weeks later the patient has scans to determine whether the TIL have successfully shrunk the melanoma. Next. When you grow the cells, you actually put them in Teflon bags, and you put them in up to 50, 60 bags in multiple incubators. And what this shows is a photograph of the inside of this incubator, which is made of stainless steel. And on the shelves in the incubator are all these bags which hold a gallon, sometimes half-gallon worth of liquid in which the cells are being nourished. So you have all these incubators filled with, 5 dozen bags, and it's, as you can imagine, a hugely labor-intensive, difficult, and expensive process. And it requires a lab that must be certified. And here at Moffitt, it took us 2.5 years to get this done right, to the point where we felt comfortable treating patients safely. Next. And the good news is that it works. If you'll pardon the disgusting photos on the right, which show necrotic tumors in the skin, and the lower ones are pre and then post. So left is pre, right is post, and the lower one is actually a giant mass that's in the underarm of the patient. And both of these masses completely shrank away 3 months after getting these TIL. And this slide summarizes a small study that was done at the National Cancer Institute by Dr. Steve Rosenberg of 35 patients who received their tumor-infiltrating lymphocytes with IL-2 after they had this lymphodepleting regimen, that is, the 2 chemo drugs to wipe out the lymphocytes. And what did they have? 3 complete responders, 15 partial, 51% response rate, and the duration of response was about a year, which is very favorable. And in some of the patients, you could see the cells in the bloodstream beyond a year, which is a phenomenally long period of time. And a lot of these patients clearly got benefit from this treatment because the ones who had a complete response stayed there for years. Next. Some of the responses were very dramatic. If you look at the left-hand side, it shows a CAT scan of a liver riddled with metastases. The patient at day -45 had their harvest. They then had a CAT scan at day -25 showing it was obviously getting worse. And then they got treated at day 0. A little over a month later, almost all gone. 18 months later, still gone. So this is a patient who eventually had a complete response and stayed there and has stayed there for a very long period of time. Next. This is one of our patients, and this is a patient with multiple lung metastases. Those white dots or circles where the red circles are all the metastases in the lung. And in one shot here, you see 3 metastases, 1 on one side, 2 on the other. And the pre means before treatment. Post is some 6 weeks later. Post number 2 is another 6 weeks beyond that. And you can tell the patient has had almost a complete disappearance of all their pulmonary tumors. Very impressive. Next. And if you look at how this treatment is done in larger numbers of patients, so again, now you're getting into about 100 patients. If you look at the curves, the blue curve shows what we do here at Moffitt. NMA means non-myeloablative, which means that you give chemotherapy to get rid of the lymphocytes, but you don't kill the whole bone marrow. At the National Cancer Institute, when you escalate what you do to wipe out the bone marrow by giving chemo and radiation, bizarrely enough, it actually looks like the patients do better, as shown in the yellow and the gray lines. And if you look at the right, the response rate of giving TBI, which is total body irradiation, with the chemo, with the TIL, with the IL-2, the whole shebang is 72%, which is a phenomenal response rate. To be honest, giving radiation is very toxic in addition to the chemo. And here at Moffitt, we've had a 50% response rate giving chemo followed by the TIL, followed by IL-2. Next. The factors that concern whether you're going to do well or not do well with this treatment can be very simple. This is a slide from a colleague of mine from MD Anderson. His name is Laszlo Radvani. And MD Anderson is another institution where they do TIL therapy as well as ours, and the National Cancer Institute, which to my knowledge are the only institutions in this country that do TIL therapy. And it shows that if you look at the patients who responded on the left in blue and those who did not respond in yellow, the number of cells given was clearly higher in the group that did well than in the group that didn't. So something as simple as the number of cells you administer determines how well the patients will do. So that means that we give never less than 50 million cells. So very simple, very straightforward determination of who's going to do well. Next. So those are TIL, and we have a number of ongoing trials planned and maybe one just about to end with TIL at our institution. But that's not the only way you can give cells for what we call adoptive cell therapy. You can also try to grow the cells from the patient's blood. Now, admittedly, most of the cells in the blood are not tumor specific, whereas many of the cells in the tumor, for obvious reasons, are tumor specific. So the idea of growing the TIL was to enrich for the tumor specific cells. But you can also get them out of the blood, and this is done at the University of Washington by my colleague Cassian Yee, who has kindly given me some of these slides, by the way. And you can actually purify T cells from the blood. And what you do is you have to amplify the cells to big numbers. And what you do is you repetitively expose the T cells in the peripheral blood to proteins from the tumor called tumor antigens. And in that way, the investigators at the University of Washington in Seattle found that they could expand the tumor-reactive population. And what they do is they put the cells into these little plastic plates in an incubator. And over time, they grow them to large numbers. And then they, just like the TIL, put them in a bag and you inject them intravenously. In this case, they give very little doses or low doses of IL-2 just to keep the cells going. And they've shown that this also can work well in patients with melanoma and other cancers. Next. It just shows the cartoon of what they do at Seattle. They actually bleed the patient at the very beginning where that green arrow is, and they collect a blood sample, and they amplify the cells over time. Over a 6 to 7 week period, when they amplify them, and they know they have enough cells growing, in the blue arrow, they just give a single dose of chemotherapy. A little less than we give with our TIL, but it still accomplishes the wiping out temporarily of the white cells. They then administer the cells back, which is that little red bag that goes into the patient. And then over time, they give, it says LDIL2, that's low-dose IL-2. And those little curving arrows are the blood draws where they determine how well the cells live in the body. And if they give low doses of IL-2 over about a month, you can actually make the cells survive over several months, and you can even induce shrinkage of tumor. Next. And it just shows a graph from a clinical trial publication which shows that when you transfer the cells that were grown outside the body, they can hang out in the body for a long time. At day 50, almost 10% of the cells are the ones that were transferred. At day 120, 5%. And up to day 220, you can still see 1% of the cells be the tumor-specific cells. And they actually change identity. If you hit next, and then next again, if you analyze the identity of the cells that you find at the time you transfer them, and almost a year later when you analyze them while they're circulating in the blood of the patient who did well, they change identity, and they become what we call memory T cells. And that's what you want. You want the body to remember the tumor so that anytime that tumor starts to grow, the cells can kill it. Next. And you can have tumor shrinkage with this treatment. This shows what we call a PET scan. And if you look at the top center of the cross-section of the body, you see a bright yellow circle. That's a tumor in a lymph node. And that's the pre-treatment sample. In the sides, the crescents are the kidneys. That's normal. So if you go to the next slide, that shows the post-treatment result. That area where that lymph node was is gone, and the yellow uptake is no longer present, suggesting the tumor is completely gone. So these T cells that are grown out of the blood can actually cause significant shrinkage of melanoma. Next. And finally, that's not the only way to transfer cells. So you can grow cells from the tumor. Those are the TIL. You can grow cells that we call clones, which are highly purified cells out of the blood. I just showed you those. The ultimate way to grow the cells is just to take the blood, and instead of growing cells that are specific for the tumor out of the tumor, which is very difficult, and instead of expanding them from tiny numbers in the blood to big numbers, you actually take the genes that identify the tumor-specific cells, and you stick the genes into the peripheral blood cells. That's called gene therapy. And this is one way to do gene therapy for melanoma. You're not putting the genes into the tumor. You're engineering the immune cells so that their ability to recognize the tumor is now changed. And if you look at the little diagram, you have the patient, the little figure of the person at the left. You isolate the rare cell that recognizes the tumor, and you take the gene that causes the recognition by the T cell, the so-called T cell receptor. You clone the gene, and if you follow the arrows, you transfer the T cell receptor gene into the regular blood cells. And then you expand those cells only over about a week or so. And you get tons of cells that are now tumor-specific. And you infuse them into the patient on the right, and you can get tumors to shrink. So this is now an off-the-shelf way to generate these killer cells. So go to the next. And again, just summarizes what I said. The gene for the receptor is cloned. The DNA from this receptor, which has two genes actually, it's what we call a two-chain receptor, goes into a virus that only replicates once, so it's a harmless, inactivated virus. You actually use the virus. Its one-time only infection is used to infect the T cells from the blood. The T cells don't make the virus because the virus doesn't replicate. They just receive the transferred gene since the virus doesn't divide. These are gene-engineered cells, and you expand them to large numbers in plates, and then you put them in these bags like I showed you. And after sufficient expansion in the incubator, just like in the picture I showed, you collect them and inject them just like the TIL. And what you do is you lymphoid deplete the patients with that chemo, then you inject the cells. So if we go to the next one, it just shows a couple of snapshots indicating how it's done. On the right side, it shows that you collect the lymphocytes from the blood, and those little round things are the plates in which you do the transfer of the gene, and then you expand the cells. You then put them into the bags where it says cell culture, and eventually you administer them into the veins of the patient. Go to the next. It just shows that these cells too can cause shrinkage of melanoma. If you look at the upper left, it shows a liver metastasis that shrank away. That's the black area and the light gray in the liver. And if you look at what is called panel C, and this is from a publication from Rick Morgan, who's at the National Cancer Institute, it shows where the arrow is before the treatment. It says -5 months. It's a mass in the lung. It got a lot bigger by day 0, and it shrank away almost completely by 2 months and 12 months. Go to the next. You can also see all kinds of different T cell receptors cause shrinkage of melanoma and other tumors when these T cell receptors are transferred into peripheral blood. This shows a clinical trial of patients who had a soft tissue sarcoma, which is a tumor of the soft tissue. And if you look at the upper panel, left is pre, right is post, and you see these big tumors where the arrows are, almost all completely went away. If you look at the lower panels, left is before, right is after. Look at where the arrows are in the liver. You see all these metastases. Again, they almost all went completely away. So it's not just melanoma that you can transfer cells in, it's also sarcoma. Next. So where do we go? Well, we know that these antibodies can treat cancer in some patients. We know that cytokines like IL-2 can treat cancer in some patients. And I hopefully just convinced you that the adoptive transfer of these immune T cells can treat cancer in some patients. What you want to ultimately do is to engineer the cells so you don't have to laboriously expand them out of the tumor or out of the blood. You just re-engineer the peripheral blood, do a brief expansion, and then boom, transfer them back into the patient. Next. So what do we know about how to do this? This is just a figurative explanation that shows how our understanding of the immune system has changed in the last 20 years. It is a rough assessment of how many articles in the field of tumor immunology have been published over those years. It's pretty obvious that the last year was hugely much more than 10 years before. Next. With that new knowledge, we hope to make better therapies. But keep in mind, the melanomas are pretty clever. They devote a large amount of their energy to avoid immune detection. They use essentially an invisibility cloak to avoid detection by decreasing the expression of these antigens. They use almost a force field. They secrete substances that actually suppress immunity. And all these things prevent the full elaboration of an immune response. And it's not easy to convince the immune system to turn against itself, because the tumor looks so much like your normal tissue, and it masquerades as normal tissue, that by provoking immunity against melanoma, you're almost asking the body to have its immune system turn on itself. Next. But keep in mind, the melanomas are darn clever. They exploit the fact that the immune system is actually hardwired to turn itself off. Keep in mind, anytime we have an immune response, like if we get influenza, we would have a vigorous immune response to influenza, but eventually it has to shut off because we're going to get better and recover. So the immune system has a yin and a yang. It has positive controls, but it has negative controls. Like a car, it has an accelerator and a brake. So all these immune responses are followed by natural brakes to turn off the immune response, so you don't all die of autoimmunity. And it essentially, the tumor coerces our own immune cells to suppress themselves by exploiting the brake that naturally exists in the immune system. But we think we can figure out a way around it. Go to the next. So how do we circumvent this? Next slide. What we do is we combine our adoptive therapy with other maneuvers that eliminate the immune suppression. And we can do that by stepping on the gas, vaccines, by giving our T cells cytokines, or we can cut the brake. And cutting of the brake, the first drug that cut the brake that was approved was the anti-CTLA-4, because immunity is like being on a seesaw. And the seesaw has to always move to the right to provoke an immune response. Next. So again, you can move it to the right by stepping on the gas, or you can keep it moved to the right by cutting the brakes. And those are the things we need to combine with our T cells, with our adoptive therapy. And as an example, our colleagues at MD Anderson and we are going to be starting a new trial soon where we cut the brakes with the ipilimumab and we give the TIL, both. Next. So what are the conclusions? I give you a positive message. Things are getting better. We know that the immune system can be a natural defense against the development of and the spread of melanoma. But the melanomas are pretty darn clever. It's a formidable foe with multiple ways to evade and avoid the immune response. We've had several decades of clinical trials that show that, yes, you can mobilize the immune system against melanoma, and sometimes dramatic tumor aggression can be seen. But we have a whole decade of basic science discoveries that show us that we have a next generation of therapies, that the next step will be the use of these TIL and the use of the gene-engineered T cells, and we're going to use them in combination with substances that promote immunity or cut the immune brake. I think we've got a bright future ahead, and I think we have time to entertain some questions.
Muted. Wonderful. We have a lot of questions for you, Jeff, from our forum participants mostly, and I have a few of my own mixed in here. So I wondered, first of all, what came to mind is melanoma any smarter than the other cancers? You talk about how it can avoid detection and so on in growing its tumors, but is it that far different than breast cancer or some of the others?
I think melanoma is more diabolical and much more clever in the way it approaches immune suppression. Melanoma has always been regarded as an immunogenic tumor for various reasons, but I think it's also evolved more ways of avoiding the immune system because of that. So I would say yes, and we're ascribing human aspects to this tumor, by the way. It's maybe a bit silly, but they are very diabolically clever tumors. I'll leave it at that.
I know Dr. Gary always says that it misbehaves. Melanoma misbehaves.
That's a good way to put it.
Quite a bit. Yes. So some very specific questions about the TIL therapy. How can you determine who will benefit best? I mean, what special criteria do you use to pre-screen patients?
The most important pre-screening characteristic is A, can they receive high-dose IL-2? So someone with cardiac disease or who's over the age of 75 would generally not be in a position to get IL-2. And since IL-2 is an integral part of the regimen, that's one eligibility. And the other is someone with exploding, rapidly progressive cancer isn't going to wait the 5 weeks to get the cells to expand and grow. It's just not practical. So the second big filtering mechanism is to decide whose disease is growing slowly enough so that they're going to be in a position to not have severe toxicity and not have a very rapidly progressive tumor, preventing them from ever getting treated. Those are the big ones.
Okay. And what are the problems associated with growing the TILs? I know some people have gone through this and they just couldn't grow their TILs and they've had to leave the trial.
Actually, these days our therapy, our technology is so good, I'd say about 85 to 90% of the tumors that are harvested grow. Very difficult to get brain metastases or gut metastases to grow, but liver, lung, soft tissue, lymph nodes, skin, all of those will grow. And we are now coming up with the use of chemicals and antibodies to promote the growth in the test tube in vitro, and I think that'll make it a 90+% chance that the TIL will grow.
Okay. And you alluded to a therapy not using IL-2 in the TILs. I know they were using IPI, I believe, out in Washington.
Actually, it's not IPI. They were using low-dose IL-2. That is, Cassiani and his colleagues used low-dose IL-2.
Oh.
At the National Cancer Institute, they're using IL-15, excuse me, which is a substitute for IL-2. And we're going to contemplate using PD-1 antibody to substitute for IL-2, much less toxic.
Oh, you're going to, oh, I thought you said you were going to use IPI. So
oh, no, we're going to use IPI, but up the road, the next plan is to use PD-1.
I see. Interesting.
The IPI would not substitute for IL-2. It would be used in addition to it.
In addition.
Yes. Whereas the PD-1 potentially could substitute for the IL-2.
Hmm. And how long do people stay in the hospital to go through this whole procedure? How long?
In our institution, they come in just for 1 day for the Cytoxan. They go home, get their 5 days of outpatient fludarabine, and then spend about a week in the hospital.
Are they in intensive care?
They are generally in intensive care once they start the IL-2. Just to get the cells, no. Get Cytoxan, no. But when they get the IL-2, generally as a precaution, we'll put them in the ICU in our institution. And then as soon as they recover, they leave the ICU, they spend a day or so in the regular ward, and then they go home.
Okay. And do you think the TILs therapy will make it to be a standardized approach, you know, regarding cost and being approved and reimbursed by health authorities?
Well, we certainly hope so. We do know that there are a couple of companies that are going to try and commercialize this. I should give due warning that I'm on the advisory board of one of them. But there are at least 2 companies who have as their professed goal the conduct of registration trials with TIL or other adoptive cell therapy for cancer. So this will be put to the test. We sure hope it succeeds.
Right. I had other doctors say that they felt it would be too costly to become very standardized.
Well, in an era where ipilimumab for 4 doses costs wholesale $120,000, you actually can come in well below that when you make TIL.
Have you ever put a price tag on the whole 9 yards of this, if you did the whole TIL
Everything? No, but to be able to make the cells and treat the patient, I think you could probably do it for under $100,000.
And so far, has the insurance company, have they all been covering the costs of these things?
No. The patients that we treat with TIL, we pay for it with a combination of grants and charitable donations.
Oh.
I mean, the IL-2, of course, is standard of care, but it's the preparation of the TIL that's incredibly expensive.
Right.
And my good colleague, Pat Hu, is applying to the Medicare to see whether they will consider covering this. But right now, the institution pays for this.
Okay.
And the IL-2, just for the growth of the cells, plus the growth of the cells, the harvest, all this stuff costs like $30,000+ a patient.
And where would you sequence this therapy in line with the others? That we have folks that start out on Yervoy, maybe do a BRAF therapy, maybe onto anti-PD-1. Where in this line of sequencing would you put the TIL therapy?
Well, some of the longest-term survivors at the NCI have been TIL patients. That would suggest to me that there's a potential to cure patients. So I would personally treat eligible patients up front. Interesting fact is that in the NCI experience, some of the best patients have had prior ipilimumab and failed. So you wonder whether you should pre-treat with IPI, then give them the TIL with the IL-2, and do that all together in the front line. That's what we're going to try to do between us and MD Anderson.
I had heard that there was some connection there with Yervoy.
And you wonder, did the Yervoy or ipilimumab cause the cells to infiltrate the tumor better? Did they activate the cells in the tumor, which then grew better? Nobody knows. But we're going to basically test that idea in a small pilot trial.
What is the difference between the NCI TIL's approach and yours and others and the other few that are going on?
Actually, our TIL and Pat Hu's TIL at MD Anderson with Laszlo Radvani are all based on the experience of the NCI. We've tweaked a few things here and there, but basically it's the classic TIL approach. The NCI has moved on to other things. They are looking at substituting IL-15 for IL-2. They're doing gene engineering with IL-12 of the TIL. They are changing the IL-2 dose. They're doing a lot of other things. So we're doing what we would think of as the classical TIL approach. And in our experience of 10 patients, 5 responders, 2 stable, 3 progressors, which is not bad for an initial pilot trial. And all of the responders are still in remission.
And the non-responders, what do they?
1 of them has died. 2 of them have gone on to other treatment.
Okay. And if someone is rejected by NCI, might they be accepted by your institution or?
Possible. That's possible.
We had one very specific question regarding that. They were rejected because of lung metastases.
Because of lung metastases?
Mm-hmm.
Yeah, it depends. I mean, it all comes down to whether we think the patient could wait the 5 weeks to grow the cells. Obviously, you don't want someone with very rapidly progressive disease or who has such severe disease that they couldn't tolerate IL-2. That's the sort of the catch.
And they had insufficient lung function.
FEF. Well, that's a potential problem. That's an IL-2 issue, and that might simply be inability to tolerate IL-2.
Mm-hmm. Okay. And out of the study that you mentioned by Rosenberg, there were 35 patients, if I correctly remember, and of the complete responses, how many were there again?
There were 3 CRs and something like 15 partial responses.
How many years has that been?
Oh, these patients were treated 5 years ago or more.
Okay, they're like 5 years out.
Oh, yeah.
And what the rest of the trial, do you know what happened with them? the rest of the patient.
If you look at the long-term follow-up that was recently published, anywhere from something like 3 to 7 years, 20% of the patients are alive and free of disease. So that's not bad.
20%. Okay.
And free of disease. There are more of them that are actually alive with disease, but 20% of them are alive free of disease. So that's probably a 20% rate of complete response leading to long-term survival, and that's very good. And that's why we're pursuing this.
Right. And again, if in the sequencing when we were talking about it, so if someone started with the TILs and did not respond, what would you recommend they do next?
Well, it depends on the individual scenario. Obviously, they'd have the choice of IPI or PD-1, or if they're BRAF, either a BRAF-MEK combination or vemurafenib, the BRAF drug that's already approved.
Mm-hmm.
That's where you'd go next.
Okay. So to the what we're now considering the traditional line
Absolutely.
of
Yes.
therapy. Okay. Wonderful. Well, that's all of the questions I received from our wonderful patients, and I can't tell you how much we appreciate you taking time out from your busy schedule
My
for us.
pleasure.
And I hope we'll be able to bring you back again to give us an update in the near
Great.
future.
Sounds good.
Great. So have a great day, and thanks again.
All right. Thank you very much. You take care.
You take care.
Bye-bye.
Bye-bye.